All right, so welcome back. We're going to start talking today about the small intestine. So the small intestine is about 20 feet long and about an inch in diameter. Now this 20 feet is really when the muscles are relaxed, and the only time you're ever going to see all those smooth muscles relaxed is, upon death. But in general, this is how long it is. So in a living person, it's a little bit short because of muscle contraction. So what we're looking at here is the stomach, right? Then the first part of the small intestine, which is the duodenum. And then the duodenum is going to go into something called the jejunum, and that'll kind of squiggle around here. And then the last part is going to be the ileum before it actually flows into the large intestine. So basically, there's a lot of surface area, and we'll see that in a little bit, inside the actual intestines, and that's going to allow for lots of food digestion as well as nutrient absorption. The three parts, we just went over that. Again, you don't necessarily have to worry about these, the actual sizes. It's more just to give you an idea of like how big they are relative to one another. But this point right down here is very important. Nearly all the chemical digestion and nutrient absorption is going to occur in the small intestine. Okay, that's its main role. So the duodenum is going to curve around the head of the pancreas. You'll see there's sort of this C-shaped curve that it does. It goes around the head of the pancreas. We saw that a little bit a while back when we talked about the pancreas. It is retroperitoneal along with the pancreas. Retroperitoneal means that, remember, we have the visceral peritoneum, visceral peritoneum, as well as the parietal peritoneum. The parietal was the membrane that would cover the abdominal cavity. This is actually behind that membrane. Okay, some of the organs actually are. They'll even go like in and out, I should say behind the peritoneum as well as in front of it. Anyway, this is behind it. It receives stomach contents. We saw that pancreatic juice and bile. In that area, in the duodenum, is where we're neutralizing our stomach acids. Right? We saw the pancreas as well as the gallbladder and liver releasing their secretions that have a higher pH, right, to help neutralize those stomach acids. We saw that there's lots of lipase coming from the pancreas, as well as the bile salts will help emulsify fats. Again, because we're increasing the pH back to a more neutral pH, pepsin is no longer going to be working because pepsin works in the stomach at a pH about 2. And again, the pancreas is releasing lots of enzymes to help digest food. The next part after the duodenum is the jejunum. This is going to have large circular folds. We'll see these in a second. Walls are fairly thick and muscular, okay? And most of our digestion and nutrient absorption are going to occur here. In the ileum, those malts, those lymphatic tissue that's found in the mucus layer, they're going to get even larger, so they get a special name. They call them Peyer's patches. These are clusters of these lymphatic nodules. And again, you're still having nutrient absorption and digestion occurring here, but at a somewhat smaller rate than in the jejunum. It ends joining up with the cecum. That's the first part of the large intestine at something called the ileocecal junction, and there's a valve there as well. It's called the ileocecal valve. So these are those folds I mentioned just a moment ago. They're called plicae circularis. So you can kind of see this roughly kind of look. Those are actual permanent folds. So they differ from the rugae in the stomach in that the rugae in the stomach can be stretched out and flattened, right? These cannot. These are permanent folds in the small intestine. Okay, predominantly found in the jejunum, or most evident in the jejunum. They disappear by the time you get to the lower ileum. They won't be found in the large intestine either. The plicae circularis will have villi on them. They're very small. They're about 1 millimeter tall. And we'll see that on the inside is the lamina propria. And it's going to contain lots of capillaries and lacteals. The lacteals are lymphatic capillaries, so that's lymphatic tissue. And I think this makes sense, right? You're going to have these villi, these little finger-like projections on top of, covering the surface of the plicae circularis to increase surface area to, again, aid in digestion and nutrient absorption. And then on top of the villi, each villi will have many microvilli. And then those are like these cells. The cell surface has what's known as a brush border. So let's just take a quick look at that in a sec. So we have, again, the microvilli. They're increasing surface area, so they're there for absorption and digestion. These cells are also secreting digestive enzymes. So that digestion is actually occurring right at the cell surface, which makes a ton of sense because, again, if these cells have to absorb these smaller nutrients, you want to digest it really close to where the absorption is going to take place. Lots of cell division because, again, you're constantly wearing away at the cells, so you're constantly having to have new cells divide for you. And basically, these are going to be those holocrine cells. So basically, if you remember back to 201, we had merocrine, apocrine, holocrine glands, or types of glands. So the holocrine glands were the ones that had cells that divided, moved to the surface, and then just burst, right? And that's the type that these are. Right. So here's just an image of this. You're going to see some of this in lab as well. So let me just kind of take you through. We're going to start here on the upper right and kind of move toward the left counterclockwise. So here you can see the mucosa layer, and so this right here would be one plique. This would be a plique right here. This is a plique circularis, right? And then on the plique, you're seeing these little, I don't know, finger-like projections off of them. Those are the villi. Okay, so in this image here, right next to it, this whole thing right here is a plique. This is a plique. This is a plique circularis. And that's, pardon me, the proper name is plique circularis. I'm abbreviating it as plique. So that's a plique circularis, and then you're seeing these little kind of things coming off of it on either side. Those are the villi. Okay, so let's just kind of zoom in on one of these villi. So on a villus, so just one of these little finger-like projections. And that's what you're seeing here. You've got the single or simple columnar epithelium. You'll see these lighter spots, like right here, kind of here, 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 and then there. Those are goblet cells, right? Also producing some mucus. And then in here, this is the lamina propria. This is where blood vessels and those lacteals would be, okay, to help absorb the nutrients. Now, if you look at the very edge, it's kind of a fuzzy edge, and it's not just because it's somewhat pixelated. If you look at the very edge, and we're going to blow that up, what you're seeing are the microvilli on the actual cells. And then this is a blown up image of one, well, I guess 3 microvilli, what they look like. But again, that's just going to help increase the surface area for digestion and absorption. So here we're taking one more look. Here we have the mucosa, submucosa muscularis serosa. That should start sounding almost like a mantra to you by now. So what you're looking at here are the individual villus, villus, villus. Each one of those is a villi. Okay, so in this image, you do not see a plique circularis. This would have to continue on, off the screen for you to see one. But you see the individual cells. You see the blood vessels in the villus, right? Capillaries, both blood and lymphatic capillaries. Otherwise, we have the same sort of layers we've been seeing so far. We have an epithelial layer, again, simple columnar epithelium with some goblet cells mixed in. The inner loose connective tissue is the lamina propria that's helping support these blood vessels. We have some lymphatic nodules, and then again, that muscularis mucosae. So, very similar to what we've already seen. Underneath that, we have the submucosa muscularis, which we have the circular and then the longitudinal muscles, and then finally that outer serosa connective tissue. So when we take a look at these intestinal glands, we have a few different types of cells. We have these absorbative cells, so these have microvilli on them, so those are those ones, right? And that cell does just that. It's going to help digest and absorb nutrients. Showed you guys the goblet cells. Goblet cells will always do the same thing. They're always secreting mucus. We have what are known as enteroendocrine cells. So these cells are producing hormones. We've already seen some of these hormones. So secretin, cholecystokinin, that's the CCK a hormone. And then we also have GIP, that's the gastric inhibitory peptide. And it was originally called GIP, but now we know it has more of a role in regulating glucose levels. So it's been renamed glucose-dependent insulinotropic peptide, which is a much more difficult to remember name, but more accurate. So basically what it's doing is it's sensing when you have sugars in your diet, and it's helping stimulate the pancreas to release insulin. Okay, not so much inhibiting your stomach anymore. Like we now know that. And then you have Paneth cells, and these secrete lysozyme. And again, if you kind of remember back to other conversations, these lysozymes are there to help break things down, particularly like inside cells. But basically, these are there to help make sure that you're keeping your gut microbes in check, okay, and basically digesting anything that shouldn't be there. Right? We also have mechanical digestion in the small intestine, and remember, mechanical digestion is just a physical division of food. So you can see here, this is peristalsis, peristaltic contractions help move things along. But basically, this other image down here is known as segmentation. So instead of the main job moving something along, what it's doing, kind of see these alternating contractions, like it's contracting here, then the contraction moves here. So now you're kind of mixing the food until you keep doing that until finally you get more of a homogeneous mixture. Okay, kind of sloshes stuff back and forth. And again, that's just helping make sure that the food mixes very well with digestive enzymes, so everything is being, fairly evenly digested. All right, we also have chemical digestion in the small intestine. Particularly, we're going to have different enzymes for carbohydrates, proteins, DNA, and RNA. So very similarly, it's like what we saw from the pancreas. However, the one thing that's missing here, there's nothing to break down. There's no separate enzyme to break down fats. Really, that's all done by the pancreatic lipase and then the bile salts from the liver. All right, so let's take a look at carbohydrates. And I've got it written here so that it's really following the entire kind of journey of carbohydrates from once you put it into your mouth all the way, down your body. So remember, the digestion of carbohydrates starts in the mouth with salivary amylase. Right? And in the esophagus and the stomach, again, nothing much is happening there. The esophagus doesn't have any enzymes to release. And in the stomach, the salivary amylase is going to denature. However, as long as the food is staying in the fundus area of the stomach, that top portion of the stomach, it can still be active for a little bit. But, it starts in the mouth, salivary amylase, maybe while it's traveling down the esophagus, those few minutes. A few seconds, it's fine. But once it gets to the stomach, there's very little carbohydrate digestion. In the duodenum, remember, we're releasing pancreatic amylase, helps digest, continue that digestion of carbohydrates. But then in the actual small intestine, we have something called brush border enzymes. So now we're in the small intestine. And these brush border enzymes are maltase, sucrase, and lactase. So we're really down to very almost totally broken down form of carbohydrates. So these are going to produce monosaccharides. So fructose, glucose, and galactose. And this one in particular you may be familiar with. Lactase, right? You've probably all heard of lactose intolerance. So lactose is the sugar, lactase is the enzyme. What happens is some people do not produce lactase as adults, because again, in general, we have a special setup to help digest milk when we're young infants, right? Because that's going to be a predominant part of our diet. However, as we get older, most people actually transition away from milk, and cow's milk and stuff like that. So evolutionarily, we don't have a lot of lactase, and some people don't have it at all. Anyway, if you don't have the lactase, it cannot break down the lactose, like the sugar that's found in milk products. Because you don't have the enzyme, the bacteria are going to start fermenting the sugar, okay? And basically you get gas, diarrhea, bloating, all those cramps, all those kinds of things. So that's what's actually happening with lactose intolerance. Okay. So again, as I mentioned, salivary amylase stops working in the stomach. Okay. Pancreatic amylase completes the first steps of the last part of carbohydrate digestion in about 10 minutes. And then those brush border enzymes, you're seeing them right here, are going to help break those things down, afterwards, after the duodenum. And this is what I was just telling you about lactase and lactose intolerance. Okay. So carbohydrates then simply get absorbed through the cell. So here we have our microvilli, and you're seeing that there's a sodium glucose symporter. If you remember back to symporters and antiporters from 201, we talked about that. So basically this does not require ATP to move glucose into the cell. We're simply using the concentration gradient. There's lots of sodium outside the cell, very little inside to help move both of those across. Same thing with galactose. Fructose is more of a facilitated diffusion. So again, still no ATP production, but it's not a symport, just a slightly different transporter molecule. Those get brought inside the cell, and then they get passed across that surface of that cell, that brush border, into the core of the villus, into where blood vessels will be. Okay. And then you see here fructose is absorbed and then turned into glucose as well. All right, so proteins, right? The beginning of protein digestion starts in the stomach. The pepsin will turn proteins into peptides. In the pancreas, we had trypsin and chymotrypsin, which turns the proteins into peptides as well as elastase and carboxypeptidase. These will all help break down other proteins in slightly other areas, not just into peptides. It'll help break down like those elastic fibers, as well as, again, just breaks it down slightly differently. And then once we get to the small intestine, we have different brush border enzymes. We have aminopeptidases and dipeptidases, and this will break those peptides that we formed down into the amino acids. Most of the absorption is going to occur via active transport, and it's really pretty much about this is going to occur in the duodenum and the jejunum. And kind of we've talked about this in 201 for those of you guys who had me for 201. We talk about like do you remember when we talked about like how come cows are all this muscle when all they do is eat grass all day long, right? And talked a little bit about, again, there's specialized stomachs when we talked about the stomach area. But only about half of the amino acids that we absorb come from food, right? And we have to have amino acids to build proteins. So if only about half of our amino acids are coming from food, where are the rest of the amino acids coming from? And that panting you're hearing, maybe, it's my big dog coming over here and she's just panting. Very excited about the lecture. She wants to know where amino acids are coming from. All right, so I'll tell you so that neither you nor my dog are kept in suspense. They come from the gut microbes, from all the bacteria that you actually have in your body, helping you break down and digest your food. That plus any cells that are actually just being shed as you're swallowing food. All right. And then lipids, so let's just kind of go through the digestion of lipids as well. In the mouth, right, remember we had lingual lipase that is activated once we get into the stomach, right? It works better at an acidic pH. But most of the digestion is going to occur in the small intestine. Again, we're going to have bile from, or I should say bile from the liver and small intestine. That's going to help break down large fats into small fats. So that's the emulsification. That's a mechanical process. We're mechanically just physically breaking it into smaller pieces. But it'll be the pancreatic lipase that will actually take those smaller pieces and convert them into fatty acids and monoglycerides. And there's no enzymes in the brush border to help break down fats. These fats are larger molecules, so really, so what you're looking at here again is a villus, right? And then inside the villus are the blood vessels as well as the blood capillaries as well as lymphatic capillaries. And basically, smaller things, amino acids, some sugars can go into the bloodstream. However, fats need to be packaged and they travel via the lymphatic capillaries, right? And you'll see they go all the way through until they go, where the subclavian and the internal jugular meet, where they finally get sent into circulation. And then this basically describes just what I said. All right, we're going to continue our breakdown of nucleic acids. Remember, pancreatic juice contains 2 of these nucleases, ribonuclease, deoxyribonuclease. We have further digestion by these brush border enzymes. There's nucleosidase and then phosphatase, and these basically break it down to the pentose phosphate, so the sugar, the phosphate, and nitrogenous bases. So it breaks down the DNA, RNA into its constituent components. And then these things are simply absorbed by active transport. Right. Lots of electrolytes. Again, lots of this is going to occur in the small intestine. They either are going to enter via diffusion or secondary active transport. There's the sodium potassium pump. Other things passively follow, so they simply go down a concentration gradient, and there's channels, so they can just simply go through the cell membrane. And other things, again, like iron, magnesium, phosphate, will need to be actively transported in your diet. We had talked a little bit about this as well. So calcium absorption, right, especially we talked about this in 201, requires vitamin D. So that, and as well as the influences by parathyroid hormone. Parathyroid hormone will help increase the absorption of calcium into the bloodstream. But you have to have this vitamin D in order to be able to absorb the calcium, because it is going to be an active transport. You're going to have very specialized transporters in order to absorb that. Okay, and the vitamin D is the thing that's going to help with the transport. If you don't have it, it can't get from the lumen of your digestive tract into your bloodstream. Okay. Other vitamins, so fat-soluble vitamins will also be transported into lacteals. And then water-soluble vitamins simply are absorbed by diffusion. B12 has to combine with intrinsic factor. And remember, intrinsic factor is being produced by cells in the stomach. So the B12 has to combine with the intrinsic factor before it's transported into cells. It's a receptor-mediated endocytosis, so you have to have this intrinsic factor will bind to the B12, and that is what will be able to attach to the receptor to bring it into the cell. And why we care about this is you have to have B12 in red blood cell production as well. So if you do not have intrinsic factor, that's one of the types of anemia that you can have. It's pernicious anemia. Okay. Here you can see the summary of water absorption. I guess liquid creation by the body as well as water reabsorption. So all of these different things are producing some sort of secretion that has lots of water in it. So saliva, gastric juices, bile, pancreatic juices, intestinal juices. So these are the ones producing it, and then your small intestine is going to reabsorb the vast majority of that. So total ingested and secreted liquids are about 9.3 liters. Again, these are textbook numbers. Small intestine absorbs pretty much all of that except for about the last liter or so. And then the large intestine absorbs that last little bit. Okay. Then obviously you don't reabsorb everything. So you're secreting about 9.3 liters. You reabsorb about 9.2. So there's some water loss, but you have to, in defecation. All right, I'm going to stop there. Please let me know if you have any questions. See you guys next time.